Okay, hello again, BIO178. In this video, we will talk about the methods section of your group project poster. So here again is that template that I provided for you on Canvas, and here we're focusing on this materials and methods section here. And the first thing you might notice is that this section takes up an entire column of the poster. So this section is going to be longer than the section we talked about previously, which was that study site section. Okay, so for our method section, generally what we're doing here is telling and in some cases showing, so I'll talk about later how we can visualize our methods, but we're telling or showing the audience what you did. So that's the gist of what we're doing here in this section. So like in the last video, what I'll do here is talk about specifics of what you should be including in this section. Okay. So first of all, what you want to do is break up this method section for our project into two different subsections. So let me go back here for reference. So in this example, each of these headers that's in bold, each one of these is considered a subsection. So for our purposes, for our poster, our group project in this semester, we want to break that up into two subsections. That first subsection is going to be called data collection. So this is essentially what was done before you got the data. This includes information about how the samples were actually collected, any processing that was done associated with that. So here, as an example, I'm showing a visual representation of a meta-barcoding workflow. So in this class, you would basically get that sequence data, analyze it to get something like that taxonomic diversity plot. So the data collection is everything that happened before you got that data, right? So it might be how the samples were collected from the organism. It might be any processing that had to take place for those samples, extracting DNA, sequencing, using certain microsatellite primers, anything like that. So that first subsection of your method section describes overall what was done with the samples to get to the point where you got that data. And I'll talk later about where you can find that information. The second subsection is data analysis. And this here is what you did with the data, how you analyzed it. So if there was a certain R package you used or something like that. So that's the second subsection of the methods is data analysis. So for both sections, one thing we want to make sure here, so this is for both sections, one thing we want to make sure we're doing is including the appropriate level of details in both of those sections. So here's in relation to this meme I have here, we want to make sure we're not giving too many specifics. So what I mean by that is, so your file type, your data, for instance, has different file types. Like it might be a .csv file or .txt file. You do not need that type of information in your method section. So we do not need details like file type of data, the exact R code. We don't need that either. I'll talk later about how we definitely want to include the R package that we used, but you do not need to include all of the exact code that you used. And I'm mentioning this because I know for many of us, if this is the first time we're writing a poster and writing up methods and results that we did for a certain project, it can be hard to understand what level of detail to include and what level not to. So this is just some general guidelines for specifics that we don't necessarily need to include, but of course that's what the drafts are for as well, if you want to turn in a draft and get some feedback from me about whether the level of detail you're including is appropriate or not. Another thing that we want to make sure we're including for both sections, so again for the data collection and data analysis subsections, we want to make sure we're using flowcharts and or images. And this is to make sure that we're presenting our methods, in an easy to follow format. So I have two examples here. These are from posters that my undergraduate research students gave at the Society for the Study of Evolution conference a couple summers ago. So the example here on the left is an example of a flowchart. So here what she did was for her entire method section, she broke it up into different sections, had different short bullet points of text and associated images for each of the different steps along the workflow analysis. Another approach you could take that may be completely different, or you might end up doing a mix of the two, is to use images. So in this example here on the right, you can see she doesn't have a flowchart, but what she does again is she has it broken up into subsections, and there are several images, each with their own associated captions included in here to break up all of the text. So you can see here on the left, she has both that flowchart and she includes images as well. On the right, it's just images, so it's up to you, but make sure you're using some kind of visuals here in your method section to make it easier for your reader to follow along. Another thing we want to make sure we're including in both sections, as I briefly mentioned in the last slide, is we want to make sure we have figure captions for every image and figure we're including. So this, for example, here is our caption that's associated with this image, caption here associated with this graph, and a caption here associated with that image. So make sure you have a caption for every image or figure you're including. Okay, and let's talk about some specifics for what should be included in individual subsections here. So this is something we want to include for the data collection subsection. Okay, and one thing we want to include here is sample sizes for each sampling site and or species that was used in our study. So this is an example that I pulled from a BIO178 group's poster last year, excuse me. And what they did here is include a table. Again, it has a caption. So you can, I should also mention this first, when you're talking about the sample sizes that were used for your sampling site and or species, it can be easiest to put that in the format of a table. Again, a table is a visual representation that makes it easy for your reader to follow along and quickly get the information that they need without having to read through a block of text. So this is an example from the last time I taught this class. This group created a table. They had 3 different sampling sites, and they included the number of samples that they used for each one of those collecting sites. So make sure you're including the sample sizes in that data collection subsection. What you want to make sure you include in the data analysis subsection, which again is describing what your group specifically did with the data. Here you want to make sure you're including the R package and or other data analysis programs that your group used to analyze the data. So depending on your group project, again, this is going to be different for every group, but you might need to include, for example, DNA Subway, or if there's a certain R package like diversity that you used. So you want to make sure you're describing in the data analysis subsection of your method section what you actually use to analyze your data. So here's an example, and I bring this up for a couple reasons. So this is the method section that I pulled from that template that I provided to you. And I actually do not like this at all. So when I look back at this, there is way too much text here. So if you look, there are just these huge blocks of text, which are not easy for the reader to follow. So this is an example from an actual poster that I presented, but I do not recommend following what I did. I don't think it reads well. What I do think you should use as a better example, what I'm showing here on the right, is a method section that I pulled from one of the example posters that I gave you on Canvas. This is from a previous BIO178 group again, so this is from our class. And what they did here in their method section, you can see they use kind of a mix of these different techniques I've talked about. So up here, they have a flowchart with an associated figure caption. They have a table here to show their sample sizes. They have some text. One thing they could add here to make it even better is some images to break up the text even more. But you can see here by comparing these two different methods, mine on the left is just way too much text. So if you could even just put an image like this over here, this example on the right is a really good example of including text, but also including some images and flowcharts to help the reader follow along. Okay, so we know now what we want to include, so how do you actually go about creating this section for your group project? Like I talked about before, the first thing you want to do is review the information I gave you on Canvas, especially for that data collection section where you're talking about what was done before you got the data. I have information for each of you on these respective pages that gives some supplementary methods to describe what was done in terms of how the data was collected, what particular markers were used. I have some publications for some of you, so make sure you review the information on Canvas. That will tell you the information you need to write, again, specifically that data collection subsection. For creating your flowchart, there are a couple of different ways to do this. This is an example of a flowchart that I created by hand for several different talks that I've used. And so what I mean by doing it manually is you can create these different shapes and then text boxes. You can do this in PowerPoint. So again, this is what I would consider the low-tech way of doing it, just by brute force creating these different shapes. So that's one way to do it. Another way to do it, here on the right, I'm showing an example from one of my previous undergraduate research students, and she used PowerPoint, but she used the Smart Art text tool. So this flowchart with each of these different bubbles and the arrows, that's pre-formatted within PowerPoint. So she chose which Smart Art she wanted to use and then filled in her information appropriately. So that's another way to use a pre-existing flowchart that prevents you from having to manually create each of these different boxes. And then just like we talked about before, once you have those different components, really the only thing left is to write the corresponding text. You want to, in general, try to keep that text to a minimum because you're breaking it up with flowcharts and images, and overall remember that full method section is going to take up one column of your poster. So hopefully that gives you some information about what should be included in your method section, how to go about writing it, but of course if you have questions, please let me know.